Hello and welcome. You're listening to the OriGene Technologies podcast. I'm your host, and I am so glad you're here with us today.
And I'm the expert. It's great to be here, and I'm really ready to dig into some fascinating science.
We have a really special deep dive for you. Our mission is, well, pretty specific. We're going to summarize a brand new research publication, a really cool one from the journal Nature.
And just as importantly, we're going to highlight how products from OriGene Technologies were actually critical for this scientific discovery.
That's right. The paper is titled A Missing Enzyme Rescue Metabolite as Cause of a Rare Skeletal Dysplasia.
And I know that title might sound a little dense, but trust us, the story behind it is basically a biological detective novel.
It really is. I was reading it and it just pulls you in. It starts with this medical mystery, a rare genetic disease called Catel-Manzke syndrome.
And it ends up revealing a completely new concept in how our cells work, a concept they're calling enzyme rescue.
Which is just incredible. And we should say right at the top, this research was powered by tools from OriGene Technologies. Their antibodies specifically help the researchers pin down a key suspect.
A very key suspect in this whole cellular drama.
Exactly. We'll get into the nitty-gritty of that later on, but
Yeah.
let's start with the mystery. Catel-Manzke syndrome. What are we actually looking at here?
So Catel-Manzke syndrome is a rare skeletal dysplasia. And clinically, it presents with a really specific set of features.
Okay.
You have something called Pierre-Robin sequence, which involves a smaller than normal jaw, a cleft palate, and the tongue falling backward, which can cause some serious breathing issues.
Right. But there's also something very, very specific about the hands, isn't there?
Yes, that's right. It's called Manzke dysostosis. It's this very distinct malformation where there's an accessory bone, an extra bone, at the base of the index finger.
Huh.
And along with that, patients generally have growth restriction and, short and long bones in their arms and legs.
Okay, so these very specific skeletal issues. The researchers tracked it down and they found a gene called TGDS.
But here's the hook. This is what got me. This gene looks like it belongs in bacteria, right?
Well, it's not that it shouldn't be there, but its presence was completely baffling to scientists for years. The TGDS gene codes for an enzyme that looks a lot like bacterial enzymes.
Specifically ones that make rhamnose, which is a sugar.
Exactly, a sugar that bacteria use to build their cell walls. But here's the conundrum. Humans and all mammals, we don't have cell walls. We don't make or use rhamnose at all.
So the question is, why do we have a gene for a bacterial cell wall builder? And why, when this supposedly useless gene breaks, does it cause such a severe bone disease?
Yeah, that's the definition of a cold case right there. You've got this gene that looks like it belongs in E. coli, but it's sitting in our DNA, and it's causing all these problems.
So how did they crack it? Where did they even start? They had to shift from genetics to biochemistry. They knew the gene was TGDS. They knew it was mutated. But they had to figure out what the protein, the enzyme it makes, was actually doing inside our cells.
So they did what scientists do. They broke it on purpose.
Precisely. They used CRISPR-Cas9 to knock out the TGDS gene in different human cell lines just to see what would happen. They want to know which metabolites, which chemicals changed when the gene was gone.
And I'm guessing they didn't find any rhamnose.
No rhamnose, not a bit. Instead, they found the enzyme produces this very specific and actually very unstable metabolite called UDP4 keto-6-deoxyglucose.
Okay, that's a mouthful. But what's the connection to the bones?
Well, the symptoms of Catel-Manzke, they overlap with other disorders related to glicosaminoglycans, or GAGs.
GAGs, right? These are the long sugar chains that are like the scaffolding for our bodies.
For cartilage and bone.
They're absolutely vital. And this is where the plot really thickens. It turns out TGDS has a cousin, an enzyme that shares about 25% of its amino acid sequence with it. It's called UXS1. UXS1. Okay, what does that one do?
Now, UXS1 is a well-known enzyme. Its job is to create something called UDP xylose. And UDP xylose is crucial. It is the starter molecule for building all those GAG sugar chains.
So without UDP xylose, you can't build the scaffold for your bone.
You can't. And here's the breakthrough. When the researchers knocked out TGDS, or the useless gene, they thought that UXS1 just stopped working.
What? Even though the gene for it was fine?
The gene's fine, the protein was there, but it just quit. And the levels of that critical UDP xylose, they just dropped.
So taking out the mystery gene causes the important one to fail. It's like, I don't know, firing the assistant and suddenly the CEO can't get any work done.
That is a very, very good analogy. And to understand why, we have to get into the mechanism. This is where we get to that amazing concept of enzyme rescue.
I love this part. This is the stalled car idea, right?
In a way, yeah.
Yeah.
Let's look at how UXS1 is supposed to work. It's an enzyme. It converts one sugar to another. To do this, it needs a helper molecule, a cofactor, called NAD++.
Okay, NAD plus.
NAD+ acts like a spark plug. It accepts electrons, which turns it into NADH, and that allows the reaction to happen. Usually the enzyme then recycles the NADH back to NAD+ internally, finishes the job, and it's ready for the next round. It's a closed loop.
But sometimes there's a problem.
Sometimes there's a glitch. The intermediate product, the thing that's working on inside the enzyme, sometimes it just falls out before the job is done.
Oh!
And when that happens, the enzyme is left holding the NADH, the used spark plug. It has no way to turn it back into NAD plus RAO. It's stuck.
It's stalled. It's dead in the water.
Completely stalled. It can't restart because it needs fresh NAD plus to begin, but its pocket is already full of used NADH.
And this is where our mystery gene, TGDS, swoops in to save the day.
This is the rescue. The researchers discovered that the metabolite made by TGDS, that long name UDP4-keto-6-deoxyglucose, is a structural mimic. It looks almost exactly like the piece that fell out of the UXS1 enzyme.
So it's like a replacement part.
It's a jumpstart. This rescue metabolite slides into the stalled UXS1. It tricks the enzyme into finishing the reaction, which converts that stuck NADH back into fresh NAD plus HARO. The enzyme resets, it wakes up, and it can get back to work.
That is incredible. So TGDS doesn't build anything itself. Its entire job is to manufacture one specific tool, a rescue metabolite, just to fix the mistakes of a completely different enzyme.
It's a dedicated repair crew. And this explains everything. If you don't have TGDS, you can't make the rescue tool.
Yeah.
Over time, more and more of your UXS1 enzymes stall out, UDP xylose levels fall, GAGs aren't made, and your bone development fails.
But wait, I have a question. So the problem is the enzyme gets stuck with NADH and it needs NAD plus arrow.
Yeah.
Why can't it just grab some NAD plus that's, floating around in the cell? Isn't there tons of that stuff?
That is the million-dollar question. And the answer is all about location, location, location.
Sure.
These two enzymes, UXS1 and TGDS, they live inside a specific compartment called the endoplasmic reticulum, the ER. And the ER is a very strange place chemically. It has very low levels of NAD plus R and really high levels of NADH.
So the enzyme is basically starving for NAD plus R, and the environment it's in has none to offer.
Precisely. And this is where we have to talk about the villain of the story, or maybe the reason for the story. Another enzyme called H6PD.
H6PD. And this is where our friends at OriGene Technologies enter the picture, right?
This is exactly where the OriGene connection becomes so vital. To prove this whole hypothesis, the researchers had to confirm exactly where H6PD was and how much of it was there.
They thought H6PD was creating this hostile environment.
Their theory was that H6PD maintains this high NADH environment, which is necessary for other things like processing cortisol. But that same environment makes it impossible for UXS1 to reset itself without help, so they had to visualize it.
And to do that, they used the H6PD Mouse Monoclonal Antibody Clone ID, OTI2A7 from OriGene Technologies.
That's the one. They used it for two key techniques, immunofluorescence and Western blotting.
Okay, so immunofluorescence let them actually see the protein inside the cell.
Yes, using the OriGene antibody, they could see bright green staining that confirmed H6PD is localized right there in the ER, the exact same place as UXS1 and TGDS.
And a Western blotting.
That tells you how much protein is there. It let them verify the protein was present and active. I mean, this was absolutely crucial. Their entire theory rested on H6PD creating this hostile low NAD plus environment. If H6PD wasn't in the ER, the whole idea would just fall apart.
So the OriGene antibody provided the hard evidence that this pressure on the system was real.
And then they did this brilliant follow-up experiment. They said, okay, if H6PD is the problem, what happens if we just get rid of it?
Let me guess. If you get rid of the thing creating the hostile environment.
You shouldn't need the rescue squad anymore.
And did it work?
It worked perfectly. When they knocked out the H6PD gene, removing that low NAD plus pressure, the cells didn't need TGDS anymore. UXS1 could just grab NAD plus from its surroundings because the ratio shifted. The disease phenotype in the cells vanished.
That's wild. It proves that TGDS is basically an evolutionary adaptation. We only have this bacterial gene because H6PD is making life so difficult for other enzymes in the ER.
Exactly. It's the price we pay to have H6PD doing its job with cortisol. We had to evolve a whole separate repair system just to keep the other factory machines running in that one specific room of the cellular house.
And validating that was all made possible by the specificity of the OriGene antibody. That's a fantastic example of how quality tools can drive discovery.
It really is.
Okay, so they proved the mechanism in cells, but a cell in a dish isn't a whole organism. Did they take this into an animal model?
They did. They moved from the molecule to the mouse. They created a mouse model that had the exact same mutation found in human Catel Manzke patients.
And what did the mice look like? Did they have the same symptoms?
Remarkably similar. The mice had brachycephaly, that's a short skull. They had shorter mandibles or jaws, and their long bones were significantly shorter. The phenotype translated incredibly well.
Wow.
And when they analyzed the metabolites in these mice, specifically in the brain and liver, they found exactly what they predicted. The levels of that rescue metabolite were way down, and as a result, the levels of UDP xylose were reduced.
Which connects all the way back to the bones.
Right. Low UDP xylose means the body can't build heparan sulfate properly, and it can't properly add sugars to a protein called alpha-dystroglycan.
That sounds pretty important.
It's critical. Alpha-dystroglycan connects the skeleton inside the cell to the matrix outside. It's vital for structural integrity. And without the TGDS rescue, that connection is weak because the sugars just aren't there. That explains the skeletal defects.
Okay, let's zoom out. We've gone from a rare disease to a weird bacterial gene to a stalled enzyme to a hostile environment proved by OriGene's tools, and finally to a mouse model. What is the big takeaway here?
The big picture is this new concept of enzyme rescue. We used to think of metabolism as these neat linear pathways. A goes to B, goes to C. This paper shows there are these shadow pathways. TGDS exists only to make a tool that fixes mistakes made by another enzyme. It's not a building block. It's a wrench for the mechanic.
And what's crazy to me is that this wrench, this rescue metabolite, is there in such tiny amounts.
That is a great point. The researchers note that this metabolite is so scarce that in other big studies it was probably just written off as, noise or an experimental artifact.
They're just junk data.
Junk data. But it turns out that noise is what's keeping our entire skeletal system from failing.
It makes you wonder what other junk in our cells is actually doing a vital job.
That's exactly the implication. The authors suggest there could be many other enzymes in our genome that we currently label as function unknown or evolutionary leftovers that are actually part of these undiscovered repair crews, just keeping everything running smoothly.
It really changes how you look at the genome. It's not just about production, it's about maintenance and repair.
And understanding this maintenance could open up whole new avenues for treating metabolic diseases. If we know an enzyme is stalling, well, maybe we can supply the rescue metabolite directly as a drug.
That's where this gets really exciting for the future of medicine.
Absolutely. And again, you can't make these big deductive leaps without the tools to precisely identify and locate all the players involved, like H6PD.
Which brings us back around. We want to say a huge thank you to OriGene Technologies. This paper is a true heavy hitter, and it's so cool to see how OriGene's H6PD Mouse Monoclonal Antibody played such a pivotal role in validating the core of their argument.
Reliable antibodies are the backbone of this kind of work. Being able to trust what you're seeing lets you make these huge leaps in understanding.
So here's a final thought for you to chew on today. We've talked about useless enzymes and junk metabolites. How many other mysteries are hiding in plain sight in our cells, dismissed as background noise, just waiting for the right tool and the right curious mind to reveal that they're actually the unsung heroes of our own biology?
That's the question that keeps science moving forward.
Thank you so much for listening to this deep dive on the OriGene Technologies podcast. We really hope you learned something new.
Thanks for listening.
We'll see you next time.